Good afternoon, everyone, and welcome to the ABG Investor Days. Today I have the pleasure to host the CEO of Cereno Scientific, Sten Sørensen. The stage is yours, Sten.
Thank you. So it's a pleasure to be here and to share with you the progress that we are doing with Cereno Scientific. We have this vision that we would very much like to transform treatment with pharmaceuticals for patients with rare and common cardiovascular diseases. And we believe we are pioneers in what we're doing. And I will share with you our progress, which we are very excited about. We just released our Q1 today, and you can read more in that if you're interested. So first, take a look at this picture. It's a woman. She's with her granddaughter, probably. And women are the people most affected by the rare disease of our lead project, pulmonary arterial hypertension. Life expectancy is 7.5 years mean after diagnosis. So it's a very terrible disease, has a fast progression, and it ends with death after 7 years. The current treatments are only dilating vessels. We believe we have something that will transform treatment by remodeling the vessels to the pulmonary artery. And for this person then to be able to live a better quality of life and a longer life and enjoy it. But let's go into a broader picture of Cereno, founded in 2012, based in Gothenburg, office also at Kendall Square in Boston. We have our major research programs going on in the US. We have worked together with Abbott. We have a collaboration with University of Michigan. And we collaborate with 10 institutions or hospitals for our lead project, the phase 2 trial with CS1. We have 3 programs, 2 HDAC inhibitor programs, and we believe we can untap the potential of that epigenetic modulation capacity which these programs have for cardiovascular disease, rare and common cardiovascular diseases. So we actually have invested and are pursuing a second program to our lead program, and that program is called CS014, and it is soon to enter human trials. We just submitted our file to the Swedish CTA for review and hopefully approval, and then we'll start our phase 1 trial in Q2 here shortly. Our third program is an IP receptor agonist that we have in-licensed from University of Michigan. And I'll tell you more in detail about these projects. We are listed on NASDAQ. We uplisted last summer, and we have had a fairly good development of the company and a share price currently around 1 billion Swedish. We had an Edison report coming out couple of months back, valuing the company to 2.3 billion top-line data for CS1 and pre-clinical phase 1 trial for the second program. So let's see. So these are the 3 programs. CS1 for pulmonary arterial hypertension is a reformulated drug, a repurposing into different indication. It's been in man for 60 years, this molecule, but we are pursuing it into this terrible disease, PAH. And we have reformulated an administration form, which is patented in 3 patent families. And we also have orphan drug designation protection in the US. So we are targeting Q3 for a phase 2 program with that compound. CS014, as I mentioned, we have submitted the IND to CTA, and we expect to start phase 1. And that drug is targeting thrombosis prevention without risk of bleeding, a major problem in that field. The third program is in preclinical and will be so for a couple more years. We haven't chosen indication for CS585. This is our very extinguished scientific advisory board. I have worked with several of these people before in my life, starting survival trials with new paradigm-shifting therapies, improving survivals with plus 30% in heart failure, 2 different trials, RALS and MERIT-HF. Dr. Pitt, to the left here, received the gold medal for lifetime achievement at European Society of Cardiology Congress last year in Amsterdam. Now, Deepak Bath is head of Mount Sinai, 8 hospitals in Manhattan. He sits on the board of Bristol Myers Squibb's major pharma. Gunnar used to be head of all clinical research worldwide for AstraZeneca. So we have a very good expertise in our scientific board. And when we connected with this scientific advisory board, they guided us to look broad for all the publications that has been published around HDAC inhibition with this molecule, VPA, which is in our first program. And we actually published 100 of the most important articles in the famous, high-esteemed peer journal, The Lancet, in 2021. So if you're interested, you could read more about it there. But what's so intriguing here is that in those publications that already is out there are many proof of concept studies that have documented pulmonary pressure reduction, also systemic reduction of pressure, reverse remodeling of heart and vessels, so going back to more normal, antifibrotic capacity and anti-inflammatory capacity, inflammation, which is the base of many origins of cardiovascular disease and other diseases. It's also documented, and we have ourselves documented, the capacity to prevent thrombosis, the major killer on the planet for humans, without increasing the risk of bleed. So these characteristics are embedded in HDAC inhibition with VPA, and we believe so also with our second project, CSO14, and that's why we're pursuing 2 projects here. Back to pulmonary arterial hypertension, as I said, it's a very bad disease, mostly females, and all the drugs there are dilators. They do not remodel the vessels and attack the origin of the disease. Maybe with one exception, I'll mention that later. So what happens? Well, the vessels, which normally are to the left here, has a certain volume, and the blood can flow from the right heart to the lungs, and the blood gets oxygenated, goes back to the left side, and is pumped out to the rest of the body. But what happens in this disease is that the vessel, the pulmonary artery, gets very narrow, and if it gets narrow, the pressure goes up. The right heart will have to pump against that pressure, and in turn, hypertrophies, grows, and eventually fails. So current therapies are, as you can see from the arrow here, trying to dilate this pulmonary vessel so there is more room to pump through the blood to the lungs. But that is only a temporary relief for these patients, and that's why, although the life expectancy has been improved from 2.5 to 7 years with those therapies, you're not really addressing the underlying cause, which is inflammation, fibrosis, hypertrophy of the vessel, and the heart. So that's what you'd like to do, and that's what we are aiming for with our CS1, the HDAC inhibitor, and the epigenetic modulation capacity which it has. So we have documentation that we address all of these factors, and the thrombotic events. We know that many of these patients also get thrombosis, microthrombosis in the vessels, and that is also a capacity that we have documented with CS1. So it looks to be really almost a perfect drug for these patients. And epigenetic modulation it's not genetic therapy where you cut in the DNA. You are impacting the production of the life-important proteins that the DNA is the map for. And that's why it's called epigenetic, outside of the genes, epigenetic modulation. So what do we have? We have a drug that has 3 patent families until 38 with extension possibilities. It's protected for 7 years after market approval in the US. We will embark on the European ODD. We have a proven safety and tolerance in the other disease for plus 50 years, prevention of epilepsy in both children and elderly. And it's in phase 2. There are some very exciting things that have happened over the last 9 months in our program with this, which is in phase 2. And you know phase 2 is safety and tolerance, and to some extent also finding out indications of efficacy. This is our principal investigator for the phase 2 trial, Dr. Raymond Benza. He's just a half a year ago hired to run this disease program at Mount Sinai for Deepak Bath and the 8 hospitals on Manhattan. So his opinion and ours is that you will, by adding this drug on top of standard therapy, will be able to remodel and restore both the vessels and to some extent the heart's function so the patients can live better and longer. The study is pursued over 12 weeks, and we are utilizing in this study Abbott's technology, which is an implantable device. You're putting it into the pulmonary artery, and we can measure the pressure in the artery and actually the heart's several functions of the heart whenever we want. And we're actually measuring, so it's without wires and without battery. It's just transported the technology to the doctor's computer. The patient only has to rest on a bed on top of a small plate. So we get 2,000 measurements out of each patient over 12 weeks, rather than a couple of measurements in the beginning of a trial and a couple of measurements at the end. So we think that is fantastic. We have 3 dosages. The low dose here is the lowest dose, which we used in our phase 1 trial that lowered pi in that trial and was safe and tolerated well. The mid dose is the double dose of that, and that was actually effective then in proof of concept in rats in PH. And the third dose is double of that. One should remember that the highest dose here is less than half of the highest dose for epilepsy, which is an asymptomatic disease and where it's used for prevention. And the lowest dose here is 1/10 of what's used in epilepsy. So we are utilizing low dosages for these patients. We're measuring a number of parameters in this study. And we are measuring how the patients feel, tolerance and safety. And we're measuring a number of efficacy parameters, the traditional 6-minute walk distance. We're measuring the heart's function. We're measuring structure, echocardiography, and MRI. And we're measuring biomarkers. In addition, we have a tool here which the physicians used daily in practice, which is called validated risk score. It's 10 different parameters, and by noting how well the patients do on those, you can actually predict both quality of life improvements and prognosis for the patients. So it will be very interesting to see what we are able to show with our drug on top of standard therapy. Now, what happened last 6 to 9 months has intrigued us and the physicians, our principal investigator, the scientific advisory board, but also those who are part of our study here, and Abbott. In June, one of the first investigators, first patient, she reported back, the investigator, that she had a patient that was so remarkably improved that she wanted to submit an abstract to American Heart Association. We got the information, agreed to pull it out, and what was seen was actually she was in New York Heart Classification 2, which you feel very bad as a patient. You're hard to breathe, hard to walk, etc. She got our drug on top of standard therapy, maximum therapy, 3 drugs, and she'd been diagnosed 3 years earlier, so she had probably around 4 years to live. She got our drug. After 12 weeks, her cardiac function was normalized, her pulmonary pressure was normalized, her pulmonary vascular resistance was reduced by 44%, and she was moved from New York Heart Classification 2 to 1, which means she was basically not feeling her disease anymore. So that was remarkable. And we decided then with Abbott and with the study coordinators that we wanted to pull out information mid-time in the study, which we did. And we communicated this on 13th of October to you, the shareholders. And what we found there, we only looked at cardiomems blindly, so pressure measurements. And we saw that 60% of the patients that we looked at had a clinically significant reduction of pulmonary pressure. Some had better reduction than the first patient case that I just mentioned. So that gave us some more confidence and excitement. And then in November, another investigator contacted us and said, you know, I don't want to let go of your drug. The patient is done with the trial, but the patient wants to keep the drug. I want to keep the drug. Can you please pursue compassionate use? So we did that with FDA, and we got that approved on January 30th. And then we asked the investigators, how many patients do you think would like to have the drug and where you would also like to continue to use it, despite that we are maybe 3 to 5 years from market? And we got back 2/3 of the patient. That was the estimate. We think maybe a half of the patients will be on long-term use, and I'll tell you a little more what that means. So compassionate use is actually something that when the physician deems that it's worth continuing to use a drug, even though it's not approved, that's the criteria. And that's also the criteria when FDA accepts that you do that. So, yeah. So what actually happens when the company pursue compassionate use or expanded access, is the name in US. You pursue a protocol. So a protocol was approved by FDA, suggested by us and approved. So now the physician and the patient is going into another trial, actually continuing from the first trial, 003, to 004. And here you can see we will measure many things over this period. And it will be minimum 1 year per patient, maybe 2 years, we'll see. And you can compare that to the 12 weeks only we have in the trial. So we will have a very good opportunity to look at many things here. Now, quickly compared to the recently approved sotatercept, which Merck bought for $7 billion a couple of years ago. We looked very similar in preclinical work. So sotatercept, Merck claims to have remodeling capacity, not really approved as such, but has been approved based on improvement for the patients. And we look very similar, use patent, impact on remodeling in preclinical work. But when you look at the side effect profile, there has come some interesting things around sotatercept with bleedings, in gastrointestinal bleedings, hemorrhage in the brain, immune reactions to the drug, and it's an injectable every 3rd week. Compare that to a drug that's oral, which is ours is going to be, and that's been in man for 50, 60 years. So we believe that we'll be quite a strong contender when we get to the market, even compared to this drug that is forecasted to 3 to 9 billion peak sales. Now, so back again, so this is the drug that we are pursuing. I'll leave CS1 there because I know I have 6 minutes left, and move on to our next program, and that's CS014. So this is a modified VPA. This is a modified of the first molecule. We have deuterized. We've taken away 2 hydrogens from this VPA molecule and pursuing that into CS014. We've gone through 5 years development program of this, and I think between 2,000 and 2,500 pages were submitted to the Swedish MPA for approval to go into man a little more than a month ago. And we believe we'll get acceptance to go into man and be able to start in June. We have contracted a CRO in Uppsala, which we've worked with before. So we'll hit the ground running, we hope, here in Q2, and aim to get that done within a year. Now, that is being developed for thrombosis. So we have thrombosis prevention data with CS014 without increasing bleeding. Bleeding is experienced by 60% of the patients who get these drugs that are on the market. All of the drugs on the market cause bleeding, and they can't be combined. And this is the major killer of man on the planet. So you'd like to have something that doesn't cause a deadly bleeding. 3 out of 100 die of bleeding because of the therapies. So this is how it can look like if you don't die. And so a new strategy is needed here, and we believe that we have that with HDAC inhibition. We are improving the endogenic defense mechanism against thrombosis, and I don't have time to go into it today, but it looks very promising, and we're doing that without causing bleed. Here you can see mice where you create actually a thrombosis by laser hurting a vessel, and if you have CS014 in advance, you see on the lower row here that you don't get thrombosis, and that's the green and the red, and we actually impact both platelets and fibrin, the glue for thrombosis. So it's very exciting. And moving on to the next and the third program, and that's CS585. So that was invented by Mike Hollinstadt at University of Michigan. We have taken and agreed on full commercial and development rights, and we are pursuing that. And that research was published in Blood, which is the premier peer-reviewed journal of blood diseases, including thrombosis, in the world. They covered it. It was published in the fall. They produced their own editorial and a podcast because they think it's so exciting that there is a new target to address thrombosis prevention with IP receptor agonists. So this is how we look at our portfolio. We think that we have a very interesting pulmonary arterial hypertension program in CS1 with epigenetic modulation. And you've seen some of the data that we have gotten a glimpse on. So in Q3, which we are holding firm to deliver the top-line data, we'll be able to share more information and see it ourselves. We believe we have a portfolio of HDAC inhibition because we have 2 programs. And CS014 actually got patent in the UK earlier this year. So very happy about that. And then we believe we have a thrombosis prevention portfolio in CS585 and CS014. And of course, if you are developing a biotech, it's good to have a portfolio. And then what do you do with that? Well, you can pursue it to market if you want to, if that's your strategy, especially for orphan diseases. But most likely and most common is that you do a deal with big pharma. Either they buy an asset or you do a co-development or licensing deal, which you see done most often. And then, of course, the company can get bought, and that happens also. So we'll see what happens with Cereno. We are pursuing both partnering and financing in parallel tracks, as always is done. Here are the upcoming milestones. So phase 2 data in Q3. Before that, hopefully acceptance of CS014 going into man. And then we will have discussions with FDA about doing a pivotal trial for CS1, and then to be able to get that approved. And we are preparing over the next year for a phase 2 program for CS014. That concludes what I'd like to say.
Okay, nice. First of all, thank you very much for this very nice presentation. Just to introduce myself, my name is Alexander Kramer, and I'm a biopharma equity analyst here at ABG. Let's continue with the Q&A. And my first question relates, of course, to your lead program. And I would like to ask you if you could outline a little bit like how a potential path to approval could look like which preparations you need to do still this year towards the end of this year after you have to read out, and which things you will do in 2025, and like how a potential path to approval could look like, also in terms of the timeline to approval.
Yeah, thank you. So first you do your readout, and actually there are 2 steps in a readout. The first one is top line. So do you meet primary objectives? We have strong belief we will do that. There are no serious adverse reports which we would have to communicate if there were. So safety and tolerance we believe we're going to hit. And then it's a matter of how much efficacy can we get out of this trial. And we will do top line first, and then there will be a continuous analysis, and we'll bring the whole package to FDA and have preliminary discussions with FDA, and then more firm discussions and a proposal of a pivotal trial. Now, depending on what we see from an efficacy point of view, we believe we'll see several kinds of efficacy in this trial based on the characteristic of the drug. But depending on how that will look, and also how the various dosages will look from an efficacy point of view, we'll have those discussions. And that will determine how the study design would be, how large the trial would be, and what design. And another factor that will determine it is FDA's view on CardioMEMS as a tool for a pivotal trial. We don't know that at this point. But if you look back and benchmark to what has been done to get approval of the last drug in PH, which is sotatercept, they did a 300-patient study in 50 centers globally, and they did that and then followed by a open label extension over 18 months. The open label wasn't needed, but the first trial was needed, and that was a year. So we expect to hit market by 2029 with such a program. Now, if FDA would accept a smaller program based on new ways of thinking of CardioMEMS utilization, for instance, we might do a shorter program or a smaller program.
Okay, very interesting. In the sake of time, maybe just two very quick questions. One related to your CSO14 program. You said you're planning to start the phase 1 program. When do you expect to finish this program? That's the first question. And the second question is, could you comment a little bit on the financial runway of Cereno Scientific, and what do you expect, how long the financial runway will extend?
Sure. So, yeah, I know that our team is working on answering questions from the CTA at the moment. So that's part of that process. But hopefully they will be able to answer in a good way and acceptable way, and then we'll get the acceptance. In that case, the study will start in Q2, which is very soon. We haven't given actually timelines for when that study will be done. I can give you summer next year. So that's a general target. When it comes to runway, so in the Q1, which was released, I think we had next to 50 million in the bank, and we got that excluded the TO3, which was prescribed to 97%, I think. So that brought in close to 74 million. So that rounds up to. And then we have a loan that we haven't taken out yet with Nordisk Forme on 45 million. So if you add these three up, you have around 170 million. That will take us, depending on how we run our programs, will take us into Q1, middle Q1 next year.
Okay, great. Thank you very much for coming today, Sten.
Thank you for your attention and interest.
And with that, I would like to close the session. Thank you.
Thank you.